Hi all, welcome back to my channel. Today I'm going to teach you about M phase. What is meant by the term M phase? M phase, which is nothing but mitotic phase. Mitotic phase, this mitotic phase takes place in diploid organisms. This M phase takes place in diploid organisms, and mostly this type of division takes place in somatic cells. Mitotic division takes place in somatic cells, or these somatic cells which are also called as vegetative cells. And this M phase is divided into four subphases. One is called prophase 1, another one is called metaphase, anaphase, teleophase. First prophase will be start, after metaphase, metaphase after anaphase, anaphase after is teleophase. So now let us discuss about prophase. Actually, this prophase is the first stage, and this prophase begins with a karyokinesis, which is nothing but the nuclear division, which is also called as karyokinesis. Nuclear division, which is called as karyokinesis. Actually, mitosis process begins with a karyokinesis. First, division of nucleus takes place, which is called as karyokinesis. What happened during this phase means condensation of chromosomal material takes place and centrosome move towards opposite poles. Now let us see about this. See, actually this is called as chromosome. This is the structure of chromosome and it is called as chromatin. This chromatin is decondensed form of DNA. It is the decondensed form. First interphase will be happen. First which phase happen? Interphase. Interphase after M phase. During the interphase, the nucleus, if you observe the nucleus through the electron microscope, then we are going to see decondensed form structure, which means the thread-like structure. This thread-like structure, which is called as chromatin, during the prophase, this decondensed form chromatin is changes into the condensed form, which is called as chromosome. The chromosome, which is made up of a DNA molecule. So this chromosome, which is called as storehouse of DNA. So this chromosome is called as storehouse of chromosome. And so condensation of chromosomal material takes place, and we know that centrosome or a centriole is the cell organelle which is present only in animal cells. This centrosome is present only in animal cells. So the duplication of centriole takes place in S phase. The centriole duplication takes place in S phase. This centrosome is moved towards the opposite poles during the prophase. So this centrosome, which is also called as asters. Asters, the term asters, which means star. The term aster, which means actually star. This is a centriole. This centriole is also called as centrosome. This centriole is the rod-shaped cell organelle. It is the rod-shaped cell organelle which is only present in animal cells, which plays a important role during the animal cell division. It produces a microtubules. These microtubules, which are also called as spindle fibers, Okay. cells at the end of the prophase when viewed under the microscope. So now the prophase is happening. If you see the nucleus during the prophase, at the end of this stage, what happened? The cell does not show Golgi complex, endoplasmic reticulum. These two cell organelles are absent in the cell, and also disintegration of nuclear envelope takes place. Nuclear envelope, which is nothing but nuclear membrane, disintegrate takes place, which means breakdown of nuclear membrane takes place. As nuclear membrane disintegrate, the chromosomes are enter into the cytoplasm of the cell. So this is called as prophase of a mitosis. See here how the spindle fibers are moving to the opposite poles, and see here we are seeing that nuclear envelope is breakdown. As this nuclear envelope will be breakdown, these chromosomes are enter into the cytoplasm of the cell. Actually, what is the shape of the chromosome? The shape of the chromosome is X-shaped. Coming to the metaphase. So, how can we know that the nucleus, the particular cell is in metaphase? How can we know that? So, as the disintegration of nuclear membrane takes place, the chromosomes are enter into the cytoplasm. By this we can understand that cell is in metaphase stage. And also it is the best phase to study the morphology of the chromosome. See, during the metaphase what happened? Before going to discuss about what happened during the metaphase, first let us discuss summary. So first is interphase. The first one is called as interphase. Interphase is divided into mainly three subphases: G1 phase, S phase, G2 phase. After the G2 phase, M phase which is called as mitotic phase. This mitotic phase takes place in diploid cells, diploid organism, in somatic cells. This process takes place in somatic cells. Actually, in this interphase, the DNA, which means the chromatin, is in decondensed form. During the M phase, what happened? This decondensed form of chromosome is changes into condensed form. So this is called as chromosome. This is the condensed form. And chromosome is made up of DNA, and these are called as chromatids. And this is called as centromer. And if you see the centromer, beside the centromer, semicircular structure we are seeing. This disc-like semicircular structure, which is called as kinetochore. Center structure is called as centromer. Two chromatids are attached by the centromer. Beside the centromer, a small semicircle disc-like structure which is called as kinetochore. This kinetochore plays an important role during the metaphase. So what is that important role played by the kinetochore? Let us discuss now. So we know that the centrosome or a centriole produce microtubules. These microtubules are attached to the kinetochore of the chromosome. So what happened after attaching this chromosome is moved to the equatorial region of the cell. Then all chromosomes are arranged parallel to each other. Then metaphase plate will be formed. This is about metaphase. That's it for today's session.